It's a project that started about 5 years ago, and the background in my laboratory is quite distinct from this project, right? So we primarily worked on mutagenesis and how it plays an important role in antibody production. And so at that time, I also was working on mismatch repair and how it plays an important role also in antibody production, primarily class switch recombination and affinity maturation. But I also knew in the back of my mind that mismatch repair deficiency predisposes to colorectal cancer. And I developed a model or a hypothesis about 5 years ago that gut microbes might be playing an important role in inducing colon cancer, specifically in the mismatch repair setting, right? Whereby gut microbes are producing DNA mutagens that are normally repaired by mismatch repair, but in the absence of mismatch repair, these mutagens would basically cause DNA lesions that would not be able to be repaired by the cellular machinery. And so hence you would get basically a catastrophic mutational effect on the colon cancer, on colon epithelial cells that would ultimately lead to colon cancer. It ended up being wrong, right? So, and as a result of that, we were at a crossroads in assessing whether we should continue with this project, but we had this really interesting finding that gut microbes were playing an important role in inducing colon cancer, specifically in mismatch repair deficient mice. And so we decided to continue along these lines, but really we needed to collaborate with a lot of other investigators because the area of research was really getting outside of our area of expertise. Our primary findings were that, first of all, that gut microbes play an important role in inducing colon cancer, specifically in mismatch repair deficient mice. That is MSH2 and MLH1 knockout mice. That a high carbohydrate diet is also associated with colon cancer, and we believe that gut microbes are collaborating with carbohydrates to induce colon cancer. Another finding that is associated with these two findings is that mismatch repair deficiency leads to a hyperproliferative phenotype in the colon epithelial cells, and that this hyperproliferation is influenced by gut microbes and by carbohydrates. So if we reduce or alter the gut microbes, we reduce this hyperproliferation phenotype. Finally, that basically this hyperproliferation phenotype, we have evidence to suggest that this is a pre-neoplastic state. 20% of all colon cancers are deficient in mismatch repair. So those are primarily sporadic colon cancers. About 20% of those are deficient. How they arise it's not really clear, but it's thought to be one of the earliest lesions that occur, genetic lesions that occur in the development of colon cancer, also alongside with mutations in the APC genes. There's also a hereditary form of mismatch repair deficiency. These are called HMPCC patients, and they do inherit an allele, a mutated allele, that ultimately predisposes these individuals to colon cancer. I think it's thought that HMPC patients 80% of them will develop colon cancer in their lifetime, so it's very high predisposition. I think it's very early stages to really be discussing these. I mean, our studies suggest that there is again, specifically in the mismatch repair deficient setting, that diet might be playing an important role, that it interacts with gut microbes, and specifically a high carbohydrate diet with gut microbes, and that modulating the gut microbiota might play an important role in altering disease pathogenesis. You know, there are some studies, epidemiological studies that do link a high carbohydrate diet to colon cancer. The relative risk is not very high, but I think what's important to be done in those studies too is to stratify patients based on genetic findings. And I think what our work shows is, at least specifically in mice, is that it's only the mismatch repair deficient mice are the ones that are predisposed to colorectal cancer through gut microbes and potentially through a high carbohydrate diet.